Hey there, welcome back to another Make Science Easy biology lesson. In this lesson, we are going to be learning all about the tests for different nutrients. We're going to be discovering how we can test our foods to find out which nutrients are in them. And we're going to be having a look at each of the chemical tests so that you know how to do these tests yourselves. The first test that we're going to look at is the test for starch. Now, in order to test food for starch, we use iodine solution. Iodine solution is naturally a brown or an orange colour. So when we look at iodine, normally it's brown or orange. However, if we put iodine solution onto food that contains starch, the starch will react with the iodine and it will turn it purple or black in colour. The purple will be very, very dark. But if we test food that does not contain any starch and we place iodine onto it, the iodine does not change. It remains orange or brown in colour. So, starch turns iodine from an orangey brown colour to a purpley black colour. The darker the iodine goes, the more starch is present. If there's no change in colour, then no starch is present. The second test we're going to look at is the test for protein. And in order to test for protein, we use something called Buiret solution. Now, Buiret solution is a mixture of sodium hydroxide and copper sulfate, and it is normally a blue colour. However, when we mix Buiret solution with foods that contain protein, the protein will change that Buiret solution from a blue colour to a purple colour. So purple tells us there is protein present. However, the more protein that we have present, the darker that purple colour is. So, lots of protein will turn our Buiret solution a dark purple. Our next test is the test for fat. And there are two different tests for fat. The first test for fat is that we rub our food that we think contains fat against filter paper. Now, if a food contains fat, that filter paper will become translucent. However, a wet food will also do the same thing, so we need to leave it to dry. Once it's had time to dry, if the paper is still translucent, then fat is present, so fats will permanently turn filter paper translucent. However, there is another test for fats. This time, we can test for fats by mixing the food that we think might contain fat with an equal amount of distilled water and ethanol. So we have our fat, our ethanol, and our distilled water. We shake and mix that together. And if there is no emulsion, then the mixture contains no fat. However, if we get a milky emulsion, then we find that fats are present. So an emulsion is a mixture and it looks a milky colour basically. So when we have our equal amount of ethanol, distilled water, and our food that we think contains fat, and we mix it together and we get a milky looking substance, and we know that fat must be present. Our next test is the test for reducing sugars. Reducing sugars are just a special type of sugar that reduce down into other substances, and I don't want to go into the chemistry of that too much right now, but all we need to know is that examples of reducing sugar are common sugars like glucose, fructose, and maltose, but table sugar, which is the sugar that we use in everyday life, is not a reducing sugar. Now, in order to test for reducing sugar, we use something called Benedict solution. Now, Benedict solution is normally a blue colour, and we place that Benedict solution into a water bath and we heat it with our food to about 60 degrees Celsius. We don't want it too much hotter than 60 degrees because it will no longer work, but we don't want it too much cooler than 60 degrees because the reaction won't take place. Now, when we have our food and our Benedict solution and it's heated to 60 degrees Celsius, we can look at the colour that it turns. If it remains blue, then there is no reducing sugar present. Green means a little bit of reducing sugar, yellow means a bit more, orange even more, and red means there is a very high amount of reducing sugar. So the colour that our Benedict solution goes will inform us as to roughly how much sugar there is in the food that we are testing. So blue is the lowest, red is the highest. In summary, all nutrients have either chemical or physical tests for them. These tests can be used to determine which nutrients are in our foods. Iodine turns black in the presence of starch. Buiret solution turns purple with protein. Fats turn filter paper translucent, or they can form in a milky emulsion with ethanol. Benedict solution turns green, yellow, orange, or red in the presence of a reducing sugar. Red means that there is more reducing sugar, and green means that there is less reducing sugar. So, I hope that you understand a little bit more about the tests for some of the nutrients in our food. Obviously, there are more nutrients. There are things like vitamins and minerals, and we haven't covered any of those tests here, but there are tests for these, but you don't need to know those at the moment. So, I hope I made science easy for you. Until next lesson, keep on learning.